All right, so thanks so much for the invite and for the really kind introduction. I want to thank everybody for tuning in today and also for sticking around for the second talk. I think this virtual seminar has definitely been a great silver lining of the pandemic, and I was able to share this link for the call with a lot of my friends and also with my family. So actually my parents are also on this call. So I'm going to try to do like a layman's version of explanation of tissue engineering as well to make sure that everyone in the audience is on the same page. So I'm Amanda. As James said, I'm a current PhD candidate at MIT in biological engineering. And today I'm super excited to tell you about this new concept that we've pioneered for tissue engineering, which we're calling engineering by elimination. And we have recently published a paper exploring this concept where we reduced this concept to practice with a specific use case of hepatic tissue engineering, which is tissue engineering of the liver. And that's because I'm working with Sangeeta Bhatia at MIT, and one of our expertises in the lab is with building artificial livers. So to give you kind of a taste of the clinical motivation for our work, our lab has specialized in tissue engineering in the liver, kind of simply put, just because it's a really huge clinical need for patients, especially if we tally up all of these different types of liver disease. And while it's true that in a healthy person, the liver has this amazing ability to self-heal and also regenerate, even if you go surgically remove up to 70% of its mass. For patients that have impaired livers, we need other types of solutions. And to date, the only cure that we have for liver disease to address fatality is transplantation of a donor liver. And we know that's not a sufficient solution because of the donor organ shortage. And so there's a lot of work going on in the academic and also biotech spaces, trying to build out other types of approaches that can make up for this donor organ shortage. So I'm showing a few of them here on the slide, such as extracorporeal devices, which operate more or less like an external liver. You can think of this sort of like a kidney dialysis machine, injectable cell therapies, and also more recently, implantable engineered tissues, which you could think of as like an artificial mini organ graft that we could stitch into your body, perhaps even permanently, to help replace or maybe alleviate lost organ function. So you'll notice that in each of these different approaches, I've drawn these brown cells, which are called hepatocytes. And hepatocytes are like the main operators in the liver. And right now we have to include these cells in these different types of therapies because they perform a lot of different types of specialized function that biology and bioengineering has yet to catch up to and be able to reverse engineer. And this is similar to something like if you have a heart failure, you can actually implant like a mechanical pump to circulate blood around the body. There isn't an analog like that for the liver. So let's talk about that a little bit more. So the liver actually performs over 500 interesting chemical and metabolic functions. Can kind of roughly categorize those into 5 bins that I'm showing here on the left: detoxification of drugs and also food products, metabolism, storage of biomolecules, production of bile, which can aid with digestion, and also synthesis of proteins like coagulation factors that help you clot when you get a cut. And these functions are really supported by this very interesting architecture that we see if we dive into the liver. So if you take the liver, there are several segments called lobules, which we're showing in this, this cutaway here. And if we then zoom in even further, there's these structures called sinusoids, which are shown at the bottom. And in the sinusoids, you can see that besides hepatocytes, there's all these other types of liver-specific cells, and they have a very particular arrangement, which is indicative of this native microenvironment. And when we take cells out of this native microenvironment, they actually do really poorly. So in the lab, what we can do is take hepatocytes out, isolate them, and then plate them on a tissue culture plastic dish. And when we do that, the cells don't perform well. So what does that mean? So I'm showing you 2 examples here. One is where this box up at the top, which is the morphology or how the cells normally look like that are of epithelial nature, that's lost really quickly in culture. And then another more quantitative way to look at this is to look at one of these 500 different functions. So this specific graph, which I'll keep showing different flavors of throughout this talk, is of production of a protein called albumin. So it fits into the protein synthesis bucket. And we're showing here that we expect liver cells to make albumin. They start out doing this, but they're actually not able to sustain that in culture because these cultures need to be more complex to support them. And across the history of our lab, in Sangeeta Bhatia's lab, we've engineered different types of platforms in vitro that can actually rescue the loss of hepatocyte phenotype. And it turns out that adding in every element of the native tissue microenvironment that I showed you on the previous slide is actually not necessary. And we can actually do just as good, or maybe sometimes even better, by co-culturing with this unique mouse fibroblast cell line that I'm showing here in blue. This concept is immediately relevant to a lot of other types of tissue culture as well, and other types of culture in vitro, because a lot of tissue culture has historically relied on this concept of using fibroblasts as a feeder culture for support. So kind of in broad strokes, on the left is one of our lab's favorite two-dimensional cultures, the micropattern co-culture, where we put hepatocytes and fibroblasts together in a specific structure, and we use that to study biology, drug metabolism, and even more complex things like the liver stage with specific pathogens like malaria. And building off of this, because where we're going with this is to make implantable therapies, we wanted to see if we could extend this idea of hepatocyte fibroblast interactions into the 3D space. And so I'm not going to overview too much for the sake of time, but we've also taken this into in vivo, and we've been able to create grafts that we're hoping in the future will be able to rescue liver disease. The main takeaway from this slide really is that fibroblasts are very important for rescuing hepatocyte function, and that we can engineer different ways to present fibroblast-derived cues in order to robustly rescue hepatic phenotype. And so now that I've shown you a primer on like liver-specific work and why the liver matters, let's take a step back and think about this in the context of the tissue engineering field. So what I've shown you so far actually aligns really well with how we basically think about traditional tissue engineering, because we take a recipe of cells, soluble factors in the culture media, and also a scaffold to initiate the culture. And then we do things like ask, what are the cells doing? And are the cells doing something like the tissues that we want them to behave like? And that's kind of an old way of looking at it. And you can find this concept in pretty much any tissue engineering textbook as the first paragraph, or in slide 1 of some type of college tissue engineering class. And it turns out more recently that people in the academic space are really busy with pioneering cool new ways to engineer tissues that add complexity, generally for the end goal of achieving something that has a much higher biological fidelity. So here are some examples of things that we can do. Of course, it's non-exhaustive, but just to show you what different types of axes of control we can achieve. So for one, with cells, we can add in different types of cells. We can have multicellular cultures instead of just one type of cell. In the area of soluble factors, we can kind of phase in different stages of presentation of growth factors and molecules in the media, and that can help us dictate how cells transition to different identities and states. And then thirdly, we can even engineer the scaffold that the cells sit in to be bioactive and take on different properties across space and time. And we think all of these advances are really amazing, but I kind of feel like the control of cells is lagging behind. And today we think that with the advent of synthetic biology, especially as we're moving more into capabilities for mammalian cells, we think we're ready to change that and to do something new with controlling cellular composition. And so today what I want to focus on for this talk is this new idea that we're adding into what we hope is the toolbox of tissue engineering, which is another way to control cells, by taking a multicellular mixture and then exerting control over its composition after we initiate the culture. And sort of the broad question that we were guided by here, and hopefully also something you can use as the audience, is what if we could engineer tissues by doing selective elimination of different cell populations? And so maybe a first reaction is that sounds kind of crazy, because why would we put a cell in that's important and then take it out? But if we take a step back and think about it, it's actually not that crazy. In fact, in development, removal of cells regularly occurs via something called apoptosis, which is a programmed cell death. And that's relevant to really normal types of proper tissue modeling, such as here when you're forming a hand, you initially start out with webbing between your fingers, and then that webbing is removed by apoptosis. And so let's tie this back to these hepatocyte fibroblasts co-culture systems that I introduced earlier. I think it's great to think about what can we do with the system, but it would also be really nice if there were some true value adds for this technology. And it turns out there's actually plenty of reasons why we might want to take out certain types of cells that we might not want to have there later, including that fibroblasts are actually not a very well-defined population. So if we're thinking forward to trying to make these therapies for patients, it actually really complicates QC of these products. Fibroblasts are also a very metabolic cell type, so they can hog a lot of the necessary nutrients from other cells that need it. And because they're contractile, they can also disturb tissue structure, which is something that we've studied and shown is very important in our lab. And it actually turns out that some previous work also from our lab suggests that the dependence of hepatocytes on fibroblasts could be transient. And we engineered and looked at this using this 2D platform shown in this video, which are called micromechanical combs, where we can manually control the distance between these hepatocytes and fibroblasts by pushing together with pipette tips or with tweezers. And to kind of quickly summarize that work without showing any data, we found that if we culture hepatocytes on one cone, shown in orange, in close proximity with fibroblasts in green, let them culture together for a bit and then pull them a little bit apart, the hepatocytes don't actually need fibroblasts to be there in contact. And importantly, if we just completely remove the fibroblasts, the hepatocytes did not do very well. So we thought this was an interesting finding because it shows that there might be some transient dependence of cells on each other in tissue engineered context, even though we traditionally think that fibroblasts are super important. And we wanted to generalize this sort of study to other 2D systems that have different configurations, not just these cone structures, and also to 3D cultures. And you can sort of understand how like these tools are not really readily available to allow us to do this in a non-invasive and also cell population specific way. And so we set out to try to make that a reality, and we landed on the idea of using an inducible suicide switch, which causes apoptosis similar to that elimination of finger webbing that I showed earlier in the introduction. We think that this approach is going to be able to help us control cellular composition. And we're calling this approach CAMEO, or controlled apoptosis in multicellular tissues for engineered organogenesis. And if you like nothing else about this talk, I hope you enjoy that we made this clever pun that we think illustrates very well that fibroblasts have a brief cameo appearance in these engineered tissues. So one embodiment of this idea is to try to employ controlled apoptosis. So we did this with a particular type of suicide switch that initiates apoptosis called inducible caspase 9. So we had our fibroblasts, which are the cells that we want to remove, express inducible caspase 9, or I'll just say iCaspase 9 for the rest of the talk, as well as the GFP reporter so we could see which cells were infected. And then we wanted to see if we could trigger that. So in an iCaspase 9 cell expressing population, shown here on the left, if you add this molecule shown in orange called CID, or chemical inducer of dimerization, it causes these unimers to dimerize, and the back end shown in blue looks exactly like the cell's intrinsic caspase 9. And that feeds into this caspase-driven pathway for initiating apoptosis, and that gets us what we want, which is cell removal by apoptosis. So we wanted to see if this works, so we took iCaspase 9 expressing fibroblasts in monoculture, added CID to see if we could trigger dimerization, and we saw that we indeed could by immunostaining for this caspase 9 dimer, which we're showing in magenta off to the right in the bottom, and that we can even see dimerization after only 15 minutes of exposure to the CID molecule. So now that we know that this works, we wanted to see, could we use this in the presence of other types of cells? And remember, the goal really is to see if we could take cells out in the presence of other types of cells. And so we used our favorite 2D culture that we showed earlier. We have hepatocytes that are surrounded by fibroblasts. I'm kind of drawing a circle here around the hepatocytes, which we've tagged the nuclei of in blue. And when I start the video, this is when we add a CID to the culture. And you can see that just the fibroblasts that are shown in green quickly bleb and undergo apoptosis. This leads to their complete removal after only a couple of hours, even at these remarkably low concentrations of CID. And sort of taken together so far, this shows us that we can engineer a way to remove a selected population of cells quickly and uniformly, even in the context of hepatic tissue engineering. So with this tool in hand, we wanted to test whether or not hepatocytes depend on fibroblast culture in these 2D configurations. So we set up a simple experiment, again with our favorite 2D co-culture of hepatocytes and fibroblasts, and then we added CID at days 1, 3, and 7 post co-culture initiation to see what would happen. And so I'm going to bring back this readout I showed from earlier about albumin secretion as a proxy for how well the hepatocytes are doing in terms of function and phenotype. And we're seeing in the dotted line where we delete the fibroblasts where the arrows are shown, that these 2D hepatic ensembles do really poorly when we take away the fibroblasts. And we think this makes sense because as the gut check I showed you earlier that with our micromechanical 2D system, that hepatocytes also did need fibroblasts. So this was kind of like a check mark for us, check mark experience for us. So moving forward, what we really wanted to do with the system is go into 3D because we're imagining engineering implantable tissues that need to have 3D structure. So we wanted to see if we could do this with our organoid-laden hydrogels. So to introduce that, what we've done is cultured hepatocytes along with fibroblasts in these microwell plates. In these microwell plates, if you culture them overnight, will form spheroids, and then you can harvest those spheroids and then encapsulate them into a hydrogel of your choice. And what we did for this particular experiment is we tried to remove fibroblasts as early as possible. So in this example, at the 24-hour mark. And by live imaging and also other characterization that I'm not showing in the slide deck today, we were able to confirm, that these fibroblasts, even in this dense cell spheroid and surrounded by a large hydrogel slab, were still able to robustly and completely remove from the system. So then we wanted to show whether or not we could see an impact on albumin function, which is that proxy we're using again for hepatocyte health. So I'll walk through this graph sort of slowly. If we have hepatocytes alone that have never seen fibroblasts before, we see really low function. And then as expected, when we add co-culture, the function is rescued. If we add a vehicle that does nothing to the system, same thing. And surprisingly, when we use our CAMEO technology to remove fibroblasts only after 24 hours of interaction, we also see no impact on hepatocyte functionality. And we wanted to really put this to the test because this is just one type of readout. So we employed another assay of liver function, specifically drug metabolism. So in this assay, what we do is we measure the expression and the activity of an enzyme called CYP3A4, and its expression is actually induced and increased whenever you add a specific drug called rifampin. And you can see in both the fibroblasts intact, which is this thing labeled vehicle, and also the CAMEO condition, which is where we take away the fibroblasts, that both of them have similar levels of increase of this enzyme expression after addition of the drug. So taken together, these two axes of liver function, which are stringent representative assays of how well the liver is doing, suggest that fibroblasts could actually be dispensable for the maintenance of hepatic ensembles in 3D, which is in stark contrast to what we found in 2D. So at this point, we were pretty excited because I think there are some really interesting applications for this, but we also wondered, why does it work this way? Why is it different between 2D and 3D? And what exactly are the fibroblasts doing? And our paper referees also wondered this, and honestly, it's a very good question, and something that we as a lab and other labs as well have tried to figure out how to answer for many years. So I wanted to share this here as a visual aid to sort of summarize what I've said so far. We know that dependence in 2D and 3D is different, and it's likely, we think, as tissue engineers because of the way that cells interact in these systems. And these interactions are actually very complicated and multifactorial. And across the literature, specifically for hepatic tissue engineering, it's been shown time and time again that trying to pick one minimal component and then present that to hepatocytes is not sufficient to rescue hepatic phenotype. So rather than take that approach, what we did instead was to try to leave the whole cell in there and then identify what might be major drivers of phenotypic stabilization, and then systematically block those to see which ones have the most impact. And so for the sake of time, I won't show all the experiments and different types of approaches that we considered, but with the context of literature, we hypothesized that some of the major differences between 2D and 3D systems include this increased amount of cell-cell interactions between hepatocytes because they're now in a spheroid. We know that happens via E-cadherin engagement, and also more hepatocyte and matrix interactions, which we know require the integrin beta-1 subunit. And in this particular experiment, to interrupt these interactions, we focused on our 3D system, which we know works, and we wanted to see if we could disrupt its stability, and essentially subtracted these specific types of interactions from culture using monoclonal antibodies that can block the binding and the specific function. And using our 2D versus 3D comparison data that I showed earlier, as well as our CAMEO system, we had already been able to identify that the first 24 hours of co-culture is an important window for looking at hepatocyte phenotypic stability. There seems to be a switch somewhere in those 24 hours. So we wanted to target these interactions pre-compaction, which is at hour 0, and also post-compaction, which is at hour 24, sort of as a control. So the last piece of data that I'll share today is that we again looked at albumin production, and in our controls, which is where we don't block anything, so there's no antibody, or we use an isotype control that doesn't interact with these targets, we see similar amounts of function, and we only see a drop in function when we block beta-1 integrin, which is the cell matrix interactions, and E-cadherin, which is the hepatocyte-hepatocyte interactions. And this impact is actually only seen when we do it pre-compaction and not post-compaction, which fits well with the idea that something important is happening and being driven by fibroblasts in the first 24 hours of 3D culture. So with that, I would like to summarize and say that hopefully we've shown you some cool things today about CAMEO as a new technology for engineering tissues by elimination. and that we have been able to use this, at least in one case, to show that fibroblasts might be dispensable for the maintenance of these hepatocytes in these specific 3D hepatic ensembles. And we were also able to use this to further guide more experiments to uncover interesting biological underpinnings. And I've shown you today the specific story with the liver, but also my awesome collaborator, Greco Song from X Chen's lab, was also able to use the CAMEO technology for engineered vasculature. And you can see, if you look at his paper in the very last figure that we put together, CAMEO are hepatic aggregates, and also their lab's engineered vascular beds, and we're able to create tissues that are also independent of fibroblasts as a multicellular system. And really kind of on a broad level, we think this is a cool new way to engineer tissues, and we could look at many different types of organs, and we think that this will be informative both for biological insights as well as future translation. And so with that, there's a lot of people to thank. They're not all on this slide, and a lot of funding sources to thank, but just for brevity, I'll highlight 4 people. I want to thank my advisor, Sangeeta, and also our collaborator, X Chen, for all the mentorship throughout this project, not just of the science, but also of me as a young scientist. And I want to thank Arnav and Greco, that are people that I'm happy to call not just close colleagues and also co-authors of this work, but also really great friends. And lastly, if you like what you heard today and would like to see my other thoughts on life science technologies and how we can have more clinical impact, I'll just quickly plug my Twitter handle. I want to thank everyone for their attention. I'd love to take any questions.
Thank you, Amanda. That was a brilliant talk. If you have any questions, please post them on the chat. I'm going to kick off. So the induction that you use, it's a chemical induction, correct? Is there any thoughts around using maybe a light induction, maybe trying to spatially control this apoptosis process?
Yeah, absolutely. We think that there's some companies where we can buy optogenetic promoters that we could potentially build into this apoptotic switch so that we can use light instead. And I think besides trying to do the spatial composition, we're also thinking about trying to put the different types of apoptotic switches that we can trigger sort of in multiplex. So maybe it's not just one cell where we have hepatocyte fibroblasts taking away fibroblasts. Maybe there's like 4 cell types, and we can really exert lots of control over even like more levels of like composition.
Okay, that was my second question. Then I think that would be really interesting to look at, actually. And then I just had a thought on, and I guess it's something which comes up in a lot of your talks. It's in terms of the release of apoptotic bodies or factors after you're stimulating apoptosis. Do you imagine, or do you ever see that there might be negative effects on the other cell type?
Yeah, that's a great question. We get that question a lot. So in our 2D system, I'll start with the 2D system. In our 2D systems, because they're removed so quickly and also they kind of release into the supernatant, we're very quickly able to remove those. And so we think there's probably not a lot of impact there. It's true that in our 3D systems, depending on what type of hydrogel you want to use and if it's going to sequester the apoptotic bodies might actually stay. And we did do some characterization as also per the request of referees to show that we do have apoptotic bodies that persist in our hydrogels, probably because we're using a fibrin hydrogel. It's more conducive to angiogenesis, which was a choice on our end, mostly for looking forward and trying to create vascularized hepatic grafts for implantation. But yeah, in a 3D in vitro culture, those apoptotic bodies are there. We've done some assays to show that when we deliver apoptotic bodies, they don't actually impact the liver function, at least for the readouts that we used. I think it would be crazy to say that they're not doing anything, but we're not seeing like huge detrimental impact. And then when we think about transitioning to in vivo, just to close the loop on all the different types of applications, we know that like macrophages will come and clear the apoptotic bodies. So we're not so much worried about the in vivo application because we think they'll get removed by like the body's like natural mechanism.
Sure. All right, thank you. And I think Natalie has some questions. Oh, actually, sorry. First of all, we have a question here from Samuel. It says, in the context of supplying hepatic tissue for transplant, do you see this process as easily scalable when thinking about manufacturing cellular therapies?
Yeah, so thanks, Dan. That's a wonderful question. It's definitely a huge thing that we think about. I think scaling is definitely not very easy. We have to think about scaling in a lot of different ways. I think in our particular regard, one of them is cell source. Like, can we get enough cells in order to have hepatic function? And are we going to use cadaveric primary human cells like we've shown in this work, or in the future maybe we'll adapt to using stem cells that we can more easily expand? And then also for scaling for our specific stuff, we need to make spheroids. So we want to think about how to expand large amounts of spheroids that still have the same uniformity and good performance that we can do at small scale. So yeah, I definitely don't think that scaling will be easy, but those are things that we're thinking about, and we have other projects that are unpublished that are exploring that.
Thank you. So Natalie, you have a question?
Yeah, you've answered one of them already, but thank you. That was about the kind of clearing by the macrophages, maybe in vivo. A second question I had was, so your data so far, especially in the 3D system, suggests that at least for the albumin function, there's a comparable response when the fibroblasts are present compared to when they're eliminated. Did you have any measures where you actually saw a beneficial effect, like greater than that of maintaining the fibroblasts when they were eliminated? And if you haven't got a measure of that now, what do you expect to see in vivo?
That's a great question. Yeah, so in our particular system where we're looking at these static in vitro cultures of hepatocyte and fibroblasts, we haven't yet seen examples of function that are better. But in Greco's paper, which we co-authored together, came out in the same issue in Advanced Functional Materials in June. We saw that when we take fibroblasts away from this hybrid fibroblast hepatocyte and endothelial cell system, that the function was actually better also when we're looking at albumin. So we thought that's really interesting that we could actually achieve better function, and we're still working on trying to figure out why that is.
Thank you. I'll pass back to James now. I think we've had another question come in.
Yeah, we've got a question from Brian, previous co-host on this series. He says, wonderful talk, Amanda. I may have missed this, but how long do hepatocytes retain their phenotype after starting CAMEO? So do you need to add or re-add the fibroblasts at a later time point?
Yeah, so that's a great question. So the hepatocytes in our in vitro culture for 3D, in the study, we deleted fibroblasts at day 1 or just after 24 hours, and then we were able to see stable function for 3 weeks. And so we haven't looked past 3 weeks yet, but there wasn't any sense of decline when we looked at albumin. So we are optimistic that it could continue to be stable for longer. And so far, we haven't had to re-add any fibroblasts at a later time point. Okay,
so that's promising. And then he actually has a second question, and so for regenerative therapies, are there risks of apoptotic bodies sticking around on your hepatocytes? So I guess that's similar to sort of the question I was asking.
Mm-hmm. Yeah, I think certainly like risks, and we have yet to really assess those things very clearly in vivo. But I think depending on, for when we're using a mouse system, if there are macrophages present, we think that they probably won't be there. There's actually this whole other axis of literature as well, which has shown that presence of apoptotic bodies could actually be used as a way to tolerize against like graft rejection and removal. So maybe like apoptotic bodies sticking around could actually be good and not necessarily a bad thing. And those questions all remain to be examined more.
All right, thank you. I think we have time for one more question. So this is from Adam, who says, do you worry about engineering tissue that is more efficient or better than the true biology? Might this introduce an improper balance when used as a therapy?
Yeah, it's a really interesting philosophical question. Yeah, it's like often hard when we're thinking about making these tissues like sort of what's like the benchmark and what's the goal and what happens if it's doing better. There are examples in some of the literature where you can engineer a tissue to overperform, and that's actually positive in some senses because you can get a small number of cells to accomplish what a larger mass of cells could do. So there's some interesting papers where cells have been modified to express, for example, coagulation factors, and you only need like 1% engraftment of those cells, and then they can rescue the entire clinical problem of not being able to clot. So I think engineering better or efficient biology does not necessarily need to be a negative thing, and potentially can actually be leveraged, especially when we go back to the first question from Sam about scaling, like perhaps it's actually a way to address scaling from a different direction.
Yeah,